Welcome to the deep dive. Imagine, for just a second, that you could walk past a champion bodybuilder, give them a high five, and instantly absorb half of their muscle mass.
Ooh, that would save a lot of time at the gym.
Right. Or maybe you bump shoulders with a brilliant mathematician on the subway and suddenly you just inherently know calculus. It sounds entirely like science fiction, doesn't it?
It really does, yeah. But the wild thing is, in the microscopic world, that exact kind of instant trait sharing happens, every single second.
And that is exactly our mission today. We are taking a deep dive into a really fascinating stack of microbiology notes to uncover where bacteria actually keep their genes and, well, more importantly, how they share them with each other.
Exactly.
Because for anyone listening to this right now who wants to understand why, say, hospital infections are becoming just completely impossible to treat, or even how scientists actually manage to genetically engineer crops and modern medicines, this is the master key.
It really is. The goal today is to completely deconstruct the traditional textbook idea of bacterial genetics. We're going to explore how scientists first figured out that DNA can physically move between cells.
Right.
And we're going to uncover why this microscopic phenomenon is, simultaneously one of the biggest global health threats we face and the most powerful biotech tool humanity has ever harnessed.
Okay, let's unpack this, because before we can understand how bacterial genes sort of manipulate the entire world, we have to understand how they're stored in the first place. And the definition most of us learned in high school biology is frankly really starting to show its age.
Oh, absolutely. It's very outdated.
Right. So to make sense of it, let's use a modern analogy. Think of a bacterium like a smartphone. The bacterial chromosome is essentially the cell's core operating system.
Yes, the OS.
Right. It's that primary essential piece of DNA that carries the genes a bacterium absolutely needs just to survive and reproduce.
Yeah, and without that operating system, the phone's just a plug. The cell dies. But then you have plasmids, and these are smaller, totally separate DNA molecules inside the cell that just replicate entirely on their own.
Okay, so if the chromosome is the operating system, plasmids are like downloadable apps from an app store.
That's a perfect way to look at it.
They carry accessory traits, right? So maybe it's an app for surviving a specific antibiotic, or an app for producing a certain toxin, or even one for digesting some really weird nutrient in the environment. They basically help the bacteria thrive in a specific niche.
Exactly. But if you delete the app, the phone still turns on, right? The cell can lose the plasmid and still survive just fine.
Right.
And that is the classic clean distinction. The chromosome is what you need to live, and the plasmid is what helps you thrive. But the thing is, what's fascinating here is that even that operating system isn't as simple as it sounds.
Yeah.
If we look closely at a bacterial chromosome, it is not just floating around freely inside the cell like, a loose piece of string. Take E. coli, for example. Its chromosome is about 4.6 million base pairs long.
Wow, okay.
Yeah. So if you fully extended that single DNA molecule, it would be roughly 1,000 times longer than the actual bacterial cell itself.
Okay, wait, that sounds like an impossible packaging problem.
Uh-huh.
How do you fit something 1,000 times longer than the container right into the container?
It requires extreme physical compaction. The DNA is what we call negatively supercoiled. So the best way to visualize this is to think of an old school curly telephone cord.
Oh, yeah, the ones that always got tangled.
Exactly. If you twist that cord just a little bit too much, it starts knotting up on itself, right? Coiling into these dense little loops to save space. Bacteria do exactly that with their DNA. They use special proteins to organize it into a dense, gene-rich body called the nucleoid.
Okay, the nucleoid.
Right. And crucially, unlike our own human cells, which protect our DNA inside a thick walled-off nucleus, bacteria don't have a membrane around this nucleoid. It is just sitting right there in the open fluid of the cell.
So we have this massive, tangled telephone cord operating system, and then these little separate circular app rings the plasmids just floating around it.
Yep.
It feels very clean, very simple, and honestly easy to memorize for a test.
It does, except the entire framework we just built, the idea that chromosomes are always singular and always circular, is completely false in the real world of microbiology.
Wait, really? So the textbook rule is just wrong?
It is entirely wrong, and it's been steadily eroding since the 1960s. Let's start with the single chromosome rule.
Yeah.
Take Vibrio cholerae, which is the pathogen that causes cholera. It normally carries its genome split across two entirely distinct chromosomes.
Two of them.
Yes. One is large, the other is smaller. But this isn't some temporary mutation or an accident. Both of these pieces of DNA carry essential genes, and both absolutely must be faithfully copied every time the cell divides.
So a bacteria can actually have a split operating system.
Precisely.
But they are at least still circular, right? Like, that's a hard rule.
Not always. Borrelia burgdorferi, which is that weird corkscrew-shaped bacterium that causes Lyme disease.
Oh, right, from tick bites.
Exactly. It has a completely linear main chromosome, like no circles at all. And it's not the only one. Streptomyces, which are these soil bacteria famous for producing like a huge portion of our natural antibiotics, they also have linear chromosomes.
Wow, so they're straight lines just like human DNA.
Yeah, they even cap the ends of their DNA with specialized proteins, sort of like telomeres. Scientists used to think that was a feature exclusively found in complex organisms like humans.
Okay, so if plasmids can be huge and chromosomes can come in pairs or be straight lines, I mean, how do scientists even know what to call a chromosome and what to call a plasmid anymore? Where do we draw the line?
Honestly, the size overlap makes it incredibly difficult. There are massive plasmids out there called megaplasmids that actually rival small chromosomes in sheer size.
Metaplasmids, that sounds intense.
It really does.
Yeah.
For instance, there's this soil bacterium called Rhizobium millilati. It helps plants pull nitrogen right out of the air. It has a standard chromosome of about 3.65 megabases, meaning 3.65 million DNA letters.
Okay.
But it also has two massive megaplasmids. And together, those two megaplasmids carry a huge fraction of the cell's total genetic code, including genes the bacterium absolutely cannot live without in its soil environment.
But wait, if it can't live without them, doesn't that make them an operating system by our earlier definition?
Yes, and that created a massive classification dilemma for microbiologists.
Yeah.
They eventually had to coin a brand new term for this specific scenario. They call it the chromid.
The chromid, like a mashup of chromosome and plasmid.
Exactly. It's a hybrid.
Yeah.
It physically copies itself using the machinery of plasmid, but it carries genes that are absolutely essential to survival, just like a chromosome. So today, the careful definition is based on function, not just shape or size.
Okay, I see.
The chromosome houses the core genetic identity, and plasmids are the mobile elements.
You know, understanding that the line between the essential core and the mobile accessory is this blurry, it naturally makes me wonder how early scientists figured any of this out. We're talking about a time way before DNA sequencing or, advanced computers even existed.
It is one of the great historical detective stories in biology, and it really unfolded in two major steps.
Okay, walk me through it.
First, they had to prove that bacteria even had a heritable material that could move. Back in 1928, Frederick Griffiths showed that if you took heat-killed deadly bacteria and mixed them with harmless live bacteria.
The harmless one suddenly became deadly, right?
Exactly. He called it the transforming principle. He didn't know what the substance actually was, but he proved bacteria could pass traits around like trading cards.
And it wasn't until the 1940s that scientists at the Rockefeller Institute purified Griffith's mystery substance and proved it was DNA, right?
Yes. Oswald Avery and his team did that in 1944. That established DNA as the molecule of heredity. But the second big leap was discovering that bacteria don't just divide identically to clone themselves, they actually have a version of sex.
Bacterial sex.
Yep. In 1946, Joshua Lederberg and Edward Tatum discovered bacterial conjugation. They showed bacteria could physically transfer genetic material through direct contact. And by 1952, Lederberg officially coined the term plasmid for these mobile pieces of DNA.
Okay, so that's where the name comes from.
Right. And right around that same time, another scientist named William Hayes made a massive breakthrough. He characterized something called the fertility factor, or the F plasmid.
Okay, what made this F plasmid so special compared to the others?
Well, Hayes found that in certain strains of E. coli, this F plasmid physically integrated itself right into the main bacterial chromosome.
Whoa, it just inserted itself into the operating system.
Exactly. And when these specific bacteria connected to a partner to mate, they would start transferring their entire chromosome into the partner in a steady linear fashion. Imagine pulling a continuous tape measure out of a box.
Which means, wait, if the DNA is transferring like a physical tape measure, you could theoretically figure out the order of the genes by tracking how long they've been connected.
Exactly. But to do that, you need a way to instantly stop the transfer at precise intervals.
And this is my absolute favorite piece of this history, because they didn't have high-tech lasers or specific enzymes to stop it. To map the E. coli chromosome, scientists literally let the bacteria start mating, and then at specific minute marks, they dump the bacterial culture into a standard wearing kitchen blender.
It is brilliantly low-tech.
I just love that. They just flipped the switch on a literal blender. The sheer physical force of the spinning blades tore the mating bacteria apart, snapping that DNA tube instantly.
Yeah, and it worked perfectly. If you blended them at 10 minutes, trait A had transferred. If you waited and blended them at 15 minutes, trait A and trait B had transferred.
It's just wild. By using timed interruptions with a kitchen appliance, they mapped the entire E. coli chromosome gene by gene.
That blender experiment is so vital because it proved definitively that the F factor could exist autonomously as a free-floating plasmid or integrate fully into the main chromosome. It showed the world that genes were modular and highly mobile.
Okay, so the blender proved scientists could physically snap the bridge between bacteria in a lab. But out in the real world, like in a hospital drain or agricultural runoff, there are no blenders stopping this process.
No, definitely not.
When bacteria connect, that transfer just keeps running.
And that reality is the engine behind horizontal gene transfer, or HGT. Plasmids move between cells using three main mechanisms, and none of them care about species boundaries.
Right.
First is conjugation, which we just touched on. A donor cell uses a physical appendage called a pilus. It's like a microscopic grappling hook and tube to connect to a neighbor and literally pump a single strand of plasmid DNA directly inside.
And what's the second mechanism?
That's transformation, which is exactly what Griffith discovered back in 1928. This happens when a bacterial cell becomes what we call competent.
Competent, like good at its job.
Well, in microbiology, competent doesn't mean capable at a job. It means the cell's membrane becomes completely porous and permeable. It just passively absorbs naked DNA floating around in its environment, which is usually left behind by dead bacteria.
Oh, wow. So they're essentially scavenging the operating instructions from a microscopic graveyard.
That is exactly what they're doing. And the third mechanism is transduction. This involves bacteriophages, which are basically viruses that specifically hunt and infect bacteria.
Okay.
When a virus hijacks a bacterium to build more copies of itself, it sometimes makes a mistake. Instead of packaging its own viral genome into the new virus shells, it accidentally scoops up a piece of the host's bacterial DNA, or even a plasmid.
Oh, I see where this is going.
Yeah, so when that newly built virus erupts from the cell and attacks the next bacterium, it injects the previous host DNA instead of a viral weapon.
Wait, I want to pause here for a second, because I've always assumed that when we hear about the crisis of antibiotic resistance on the news, it meant a single bacteria somewhere, like randomly mutated, survived the medication, and then slowly multiplied over time.
Right, the classic Darwinian model.
Yeah, like a slow evolutionary march of survival of the fittest. Are you saying that's wrong?
It's not wrong that mutations happen, but it vastly underestimates the speed of the current crisis. A great deal of clinically important resistance, especially multi-drug resistance, travels laterally as an entire prepackaged software update.
Wow.
You aren't waiting for one cell to independently mutate and invent resistance to 5 different drugs over a million generations. The bacterium just downloads the pre-written code for all 5 resistances at once from a passing plasmid.
So it skips the evolutionary waiting room entirely.
Yes, and we've known about this for a long time. Look at the 1959 Japanese dysentery epidemic caused by Shigella. Physicians were completely baffled because they were seeing strains resistant to multiple unrelated antibiotics simultaneously.
Which shouldn't happen that fast.
Exactly. If it were just random chromosomal mutation, resistance would accumulate slowly, one drug at a time. But a researcher named Tsutomu Watanabe proved that this multi-drug resistance was transferring as a single massive package between entirely different bacterial species.
Ah, so they discovered our plasmids. Resistance plasmids.
That was the massive turning point. Plasmids went from being a quirky phenomenon in laboratory genetics to an urgent matter of global public health. It showed us that antibiotic resistance could spread suddenly, wholesale, across species.
To bring that right up to the present day, we just have to look at NDM1, because this is the modern crisis. NDM1 was first found in 2008 in a patient hospitalized in New Delhi.
Yeah, a very scary moment.
This specific gene creates an enzyme that outright destroys carbapenem antibiotics. And just to be clear for you listening about what carbapenems are, they are like a master skeleton key that breaks the cell walls of almost any bacteria. They are the heavy-duty drugs we use when absolutely everything else fails.
The drugs of last resort.
Exactly. But resistance to them didn't spread slowly, it just exploded worldwide.
And the reason it spread so fast is the core of everything you're discussing today. NDM1 didn't spread because one single resistant strain of bacteria booked a bunch of international flights.
Right.
The NDM1 gene sits on highly mobile conjugative plasmids. When one bacterium hands the NDM1 plasmid to a neighbor, that neighbor doesn't just survive the antibiotic. It instantly becomes a donor itself.
Oh, man. It's an exponential chain reaction.
Precisely. One donor bacterium infects a neighbor, making two donors. Two become four, four become eight, eight becomes 16. Because the plasmid carries the blueprints to build its own transfer machinery, a single successful mating event can convert an entirely susceptible bacterial population into an unstoppable drug-resistant army in a matter of hours.
Hours.
Yes, the resistance appeared independently in completely unrelated bacterial lineages on different continents at the exact same time.
That is terrifying. Honestly, that actually brings up a biological paradox that has always bothered me. Copying DNA takes physical energy, right?
Yes, quite a bit.
Oh. So if a bacterium has 100 extra plasmids, the cell has to spend precious energy replicating and reading all that extra code. So why do bacteria keep these plasmids around when antibiotics aren't present? If I don't use an app on my phone, I delete it to save battery. Why don't bacteria just drop the costly extra luggage?
That is the famous plasmid paradox. For decades, some scientists viewed them as just freeloading selfish DNA. But plasmids persist in the wild at incredibly high frequencies, even when they aren't providing a useful trait.
So how do they stay?
To understand why, you have to realize plasmids have evolved absolutely ruthless survival tactics. One of the most fascinating mechanisms is the use of what we call addiction modules.
Addiction modules, that sounds like a molecular hostage situation.
That is exactly what it is. Addiction modules are antitoxin systems written straight into the plasmid's code. The plasmid forces the cell to produce two things: a highly stable, long-lasting toxin and a very unstable, short-lived antitoxin.
Yeah.
As long as the bacterium keeps the plasmid, it keeps manufacturing the short-lived antitoxin, neutralizing the poison, and it survives.
Wait, but what happens if the cell divides and, forgets to pass the plasmid to its daughter cell?
Then the short-lived antitoxin degrades almost immediately. But that long-lasting toxin is still floating around in the daughter cell's cytoplasm.
Oh, no.
Yep. Without the plasmid to manufacture more antidote, the toxin becomes active and literally kills the daughter cell. The plasmid enforces its own retention with a dead man switch. It's basically saying, keep me or you die.
That is unbelievably sinister. Do the bacteria have any way to fight back against this?
They do, and it's a system you've likely heard of in a totally different context: CRISPR-Cas.
Oh, like CRISPR, the gene editing tool.
Exactly. We hear about CRISPR on the news as this revolutionary human tool. But in nature, CRISPR evolved as a bacterial immune system. Bacteria use it to specifically target and slice up foreign DNA, including invading viruses and parasitic plasmids.
So it's a war.
It's an endless evolutionary arms race. Plasmids try to force their way in, and bacteria try to cut them up. Plasmids also adapt by relying on sheer copy numbers. Some plasmids ensure they get passed on by just flooding the cell, making tens to hundreds of copies of themselves, so that when the cell divides, every daughter cell gets at least one just by statistical chance.
Understanding that microscopic arms race completely shifts how we look at biotechnology. Because human scientists looked at how effectively plasmids hijacked bacterial machinery to deliver genes and realized, well, here's where it gets really interesting. We could hijack the hijacker.
Right, we could use this natural gene delivery system for ourselves.
Exactly.
And the most elegant example of this is the T plasmid, the tumor-inducing plasmid from a soil bacterium called Agrobacterium tumefaciens.
In nature, this bacterium is essentially a rogue agricultural engineer, right?
That's a great way to put it, yeah. Agrobacterium infects wounded plants.
Yeah.
And it uses its massive T plasmid to transfer a segment of its own DNA directly into the plant's chromosomes. It actually edits the plant's genome. It forces the plant to produce these tumors called crown galls, which act as a safe home for the bacteria. And it forces the plant to manufacture specialized nutrients called opines that only the bacterium can digest.
So it genetically engineers the plant to build it a house and cook it dinner.
It really does. But human molecular biologists in the early 1980s realized what was happening. They took this natural T plasmid and they surgically stripped out the tumor-causing genes.
They essentially hollowed out the delivery vehicle.
Exactly. Then they replaced those disease genes with genes we wanted the plant to have, like, say, drought resistance or better nutritional profiles.
And just like that, the T plasmid became the foundational workhorse for classical plant genetic engineering. It is the basis for so many of the modified crops grown globally today.
Yep. But the application doesn't stop in agriculture. Plasmids are humanity's primary tool for inserting genes across almost all domains of life. If we look at that high copy strategy we discussed earlier.
Where they flood the cell with copies.
Right. Scientists took a naturally occurring plasmid lineage, repurposed its ability to make hundreds of copies of itself to maximize DNA yield in the lab, and engineered the classic cloning vectors used in biology labs all around the world.
To put that into perspective for you listening, what does this mean for us today in practical terms?
It means the entire infrastructure of modern medicine runs on plasmids. Recombinant human insulin that keeps millions of diabetics alive, many of our modern vaccines, cutting-edge monoclonal antibody treatments, and personalized gene therapies, they're all manufactured using plasmid vectors.
That's incredible.
We are utilizing the exact same conjugative machinery nature uses to spread antibiotic resistance to manufacture the medicines that keep us alive.
It is an unbelievable amount of power to hold, but, holding that power also terrified the people who invented it. We absolutely have to talk about the 1975 Asilomar Conference.
Oh yeah, it's a crucial piece of this story. In 1973, scientists Stanley Cohen and Herbert Boyer successfully spliced genes into plasmids and reintroduced them into bacteria. It was the birth of recombinant DNA technology.
But because these scientists deeply understood how easily a plasmid can carry a gene into a new host and how easily bacteria share plasmids horizontally through the environment, they panicked.
They realized the genie was out of the bottle. They were genuinely terrified of accidentally creating a dangerous, transmissible, completely unstoppable organism in the lab before they fully understood the rules.
So what did they do?
The Asilomar Conference was this landmark moment where scientists from around the world gathered and voluntarily paused their own research. They enacted a moratorium to establish strict safety guidelines. And they did this entirely because they respected the incredible, uncontrollable mobility of the plasmid.
Right. They knew that if a genetically engineered plasmid got out into the wild, the exponential chain reaction of horizontal gene transfer could spread it globally.
Exactly. It was a moment of profound responsibility.
Okay, we have covered a massive amount of ground today. Let's quickly recap the journey. We started by breaking the textbook rules. The bacterial chromosome, usually the core OS and plasmids, the mobile apps, aren't always strict, neat categories.
No, not at all.
We've got massive chromids and linear DNA blurring the lines completely. We explored how these genes move via horizontal gene transfer mechanisms so robust that early scientists could literally map them by stopping the process with a kitchen blender.
Still my favorite part.
Same. We saw how the exponential spread of these plasmids is the terrifying vehicle for multi-drug resistance in our hospitals. And yet, they are simultaneously the hijacked, unsung heroes of modern biotechnology, agriculture, and life-saving medicine.
So I guess the next time you hear a news report about a terrifying new hospital superbug, or you read a hopeful article about a breakthrough in personalized gene therapy, I want you to picture the invisible world. Picture these tiny rings of DNA constantly jumping between species, completely ignoring the boundaries of life as we categorize it.
It really changes how you look at the physical world around you.
It does. And it leaves us with one final kind of radical concept from evolutionary biology to think about. Plasmids move horizontally across entirely different unrelated species, while the host bacteria only reproduce vertically, passing traits down to their direct descendants.
Right.
When you look at that dynamic, it challenges our entire organism-centered view of evolution. You kind of have to wonder, is a plasmid just a convenient accessory for the bacterium? Or is the bacterium just a convenient temporary vehicle for a quasi-parasitic, independent replicator that has actually been running the microscopic world all along?